our first speaker is going to be Dr. Eric Brunskill. He's Program Director at the NIDDK for the Division of Kidney, Urologic, and Hematologic Diseases. He obtained his PhD at the University of Maryland. He subsequently did his postdoctoral training at Cincinnati Children's Hospital, and he went on to get his first faculty appointment there. He joined the NIDDK in 2020, if I'm correct. And as program director, he's responsible for administering a grant portfolio on basic cellular, molecular, and genetics of normal development, maturation, and aging of the kidney and lower urinary tract. So Dr. Brunskill is going to talk to us about several resources. One is the GUDMAP, as they stands for GenitoUrinary Development Molecular Anatomy Project. The other one is Rebuilding a Kidney, so RBK. And the third is Kidney Precision Medicine Project, or KPMP. So Dr. Brunskill.
Good afternoon. Again, thanks, Bernadette. Thank you for the opportunity. I'm Eric Brunskill from the NIDDK, and I'd like to really take a couple of minutes really to tell you about some of the projects that the NIDDK sponsors and some of the resources that are available to future investigators. And so I have nothing to disclose. And so just to start off with, as many of you know, the mission of the NIDDK really is to support medical research and research training and to disseminate science-based information on diabetes and other endocrine and metabolic diseases. And these include nutritional disorders, obesity, as well as kidney, urological, and hematological diseases. And the mission of our research is really to ultimately improve people's health and quality of life. And I put here, again, some websites for you. You can reach us at niddk.nih.gov or any of the other links that I have listed here for any additional information about our institute. And so, as I mentioned, the NIDDK's mission is to improve the health of individuals. And we do that through a number of funding mechanisms and research programs. And I've just listed a few here, some of the key programs that are supported by the NIDDK, and these include the RBK, the Kidney Precision Medicine Project, Neptune, the GenitoUrinary Development Molecular Anatomy Project, or GUDMAP, and the CRIC study. But as Bernadette told you, I'm just going to tell you about three of our research programs: the RBK, GUDMAP, and KPMP programs. So the first one that I'd like to start off with is the GUDMAP program, which I think everyone is in this audience is aware of this consortium, and it's been going for 17 years strong now. The resources that are available is something, again, that everyone is, I think, available, and GUDMAP continues to provide the urological community access to a wealth of information regarding development. However, there's some recent changes that you might not be aware of that I'd like to share with you today. So in the past, GUDMAP has provided, again, a wealth of resources for the urology community. And these were made available to the community through the goodmap.org website, which I think perhaps a lot of people have visited in the past. And on this site, you'll continue to find a variety of different resources, including imaging and transcriptomic data, as well as experimental protocols and tutorials on how to do experiments. These are all available through this GUDMAP resources. And the website has been created that there are multiple ways that you can search the data that are available and these resources, whether it's entering in by a gene or even a disease state. You can find the related resources through the GUDMAP website. And I'd really like to say that to keep up with the need for more computational and data sharing, the GUDMAP resources have recently been moved to the cloud with an eye towards the future on really how to share data and resources seamlessly. And so all of these resources are available on an AWS cloud, even though you won't notice it, but in the future, we'll be able to transfer data seamlessly between site to site. One of the other things I'd like to tell you about is that there are many new faces to the GUDMAP consortium. The current GUDMAP consortium is really focusing on the lower urinary tract. In the past, it's been more of a kidney-centric focus, and we have a new set of researchers from a variety of institutions throughout the country, as well as Australia, who are digging into understanding urology and especially generating new data sets that have not been available before to the urology community. And so we are generating, again, molecular atlases, which I'll tell you a little bit about in a couple of minutes, but we are generating a variety of molecular data using single-cell ATAC-seq, single-cell spatial transcriptomics. We're doing codex, nano CT imaging, as well as continuing to generate new protocols and tutorials for the community. And so these new investigators, I think, will be building a new network and add to the GUDMAP story as it continues. And one of the main goals of this new iteration of GUDMAP that we just funded this year is really strengthening community outreach. And GUDMAP will be continuing to promote interactions and fostering collaborations between basic scientists, clinician researchers, and non-traditional, renal and urological researchers interested in studying all things urology. These resources that these new researchers are developing will be some high quality 2D and 3D images and atlases of the lower urinary tract. GUDMAP Forward wants to continue to lead the field of developmental urology and utilize state-of-the-art tools and techniques to promote discovery. They also will want to generate resources for the community that will help them generate new hypotheses for them to write new grants. Most importantly, though, what we'd like this new iteration of GUDMAP to do is really engage and collaborate with the broad research community, especially the future investigators. We'd like GUDMAP to be a nucleating center for diverse and emerging scientists to present and share ideas. And so we're working on diversity supplements, opportunity pools, and collaboration with other Atlas projects to bring in young investigators into the GUDMAP fold, if you will. In addition, our community outreach, we want to continue to, again, as I say, promote and fostering collaborations between basic scientists and clinician researchers. And one of the things we've done in the past, and we'd like to continue on doing, are holding these GUDMAP jamborees, where we engage and integrate outside researchers into the GUDMAP consortium, and we broaden the urology community and allow everyone to share their ideas, again, allowing GUDMAP to be a nucleating center for the entire urology community. And so, again, I'm trying to fit 3 different talks in here really quickly. If you have any questions at any time, please contact myself or my colleague Anna Sadusky, who's the program officer for GUDMAP, about any questions you may have on training programs, grants, etc., related to GUDMAP. Now, I'd like to switch gears one really quickly and tell you about another resource that is available to you, and that's the Rebuilding a Kidney, or RBK. And the RBK is more disease focused and seeks to enhance endogenous productive repair and regeneration by small molecules, proteins, or engraftment of cells or organoids. And in addition, the RBK seeks to build functional replacement kidney tissue ex vivo for transplantation, quite different than what you're used to perhaps in urology. However, I think there are many types of data sets that are generated by these RBK researchers that you might find useful. And so again, while the RBK might be a little different than the lower urinary tract research, there are still a number of different resources available through the RBK or rebuildingakidney.org website. And these include single-cell RNA-seq data sets. There's information on funding, as well as resources for patients to stay on top of stem cell and stem cell developments. In addition, on this website, we try to communicate with the field through our Twitter feed. And so please check out the site, as you might find a number of resources useful to you, and follow us on Twitter, where they always post the latest research achievements, meetings, and funding opportunities. Again, please feel free to contact us at any time, either myself or the program officer, Danny Gossett. We are more than happy to answer any questions regarding RBK. Now, one thing I'd also like to tell you about, and one of the changes this year that you might not be aware of, is that NIDDK funded a new data hub called the D2K Atlas Center, which is an acronym for Analysis, Technology, Leadership, Administration, and Science. And the Atlas Center will continue to host the GUDMAP and RBK websites, but with a few changes. The Atlas Center really aims to go beyond the repository data structure and build an interactive resource that is aimed at being the go-to open access resource for the community, and provide an interactive knowledge base for the research community regarding mouse and human renal and genitourinary development and disease. The Atlas Center will build an interactive 2D and 3D atlases and molecular maps with FAIR principles. And most importantly, the Atlas Center will listen to the urology research community to learn how researchers would like to use GUDMAP resources. And this could be developing state-of-the-art computational tools, or allowing individuals without computational backgrounds to delve into these data sets to really delve in and generate new hypotheses. And so in this environment of ever-increasing data complexity, data analysis is really now the bottleneck of discovery. And in the upcoming months, you'll see changes to the GUDMAP and RBK websites that will help users analyze the data more efficiently and without a computational background. And the last thing I'd like to tell you about in terms of resources is the KPMP, or Kidney Precision Medicine Project. And the KPMP is a multi-year collaboration of leading research institutes that are engaged in studying patients with acute kidney injury as well as chronic kidney disease in order to understand the disease mechanisms and to develop treatment individuals that suffer from AKI and CKD. And the goals of KPMP are really to first ethically obtain and evaluate kidney biopsies from patients that have acute kidney injury or chronic kidney disease. And then the KPMP utilizes state-of-the-art molecular technologies on these biopsies in order to define disease subtypes and subgroups. Using sophisticated computational approaches, KPMP analyzes the data from these various molecular technologies and identifies critical cells and pathways that ultimately will lead to potential targets so we can develop novel therapies to treat, again, AKI or CKD. Ultimately, what this group is interested in, the KPMP curates this data and makes a molecular atlas of this data, which is available for the community to use. And so the data generated by the KPMP is released to the community as quickly as possible through the KPMP Atlas. And that's shown at this atlas.kpmp.org. And the data here is, again, available to all external researchers through the Kidney Atlas portal, where they can interrogate the data at quite a high level. The data from this research will help answer questions important to people with kidney disease, such as what type of kidney disease do I have? What will happen to me? What can I do about it? And so all this data is through a Kidney Atlas portal at atlas.kpmp.org. And again, it provides tools that allow users to, without computational background, to explore the data at quite a high level and develop hypotheses for grants and other mechanisms. And so lastly, I always like to mention the innovators pipeline, which is critical for future research endeavors and our future investigators. And so NIDDK has a comprehensive training and career development program, a suite, and that start as early as high school and undergraduate levels, and they include programs that involve individual awards or grants to institutions to manage programs for multiple trainees. While I don't have the time to cover any of these opportunities in depth here, please don't hesitate to contact myself or one of my colleagues when we'll point you in the right direction to point you to the people who you need to talk with to answer your questions on these research programs. And so we are here to help especially the future investigators find answers to their questions regarding anything about the NIH or NIDDK. And so please don't hesitate to contact myself or my colleagues, Anna Sadusky or Danny Gossett, who are also involved in the GUDMAP and RBK programs. And again, I always look forward to talking with each one of you in the future. And hopefully I did that under time.
Wonderful. Thank you so much. So we'll take some questions till the end. So if you have any, make sure you write them down or put them in the chat if you prefer. So we'll move on to our next topic. So this will be addressed by two speakers. We've got Dr. Rebecca Rodriguez. She's a repository program director at She obtained her bachelor's degree from Rider's University, her master's from University of St. Joseph, and then she went on to get a very diverse research experience in both basic and clinical science. And then she ended up obtaining her PhD at the University of Hawaii in John A. Burns School of Medicine. Don't know how you get your PhD at Hawaii because I would be sitting on the beach. And then after she obtained her PhD, she went on to join the NIH, where she currently fits in her role as program director. In addition to Dr. Rodriguez, Dr. Keller will be joining her. He's a principal director in Booz Allen Hamilton's life sciences business, and he has over 20 years of extensive experience conceptualizing and implementing large-scale research and data sharing platforms across the federal government. So this focuses on both NIH and VA. He holds a PhD in microbiology and immunology that he obtained from Wake Forest University School of Medicine. So together they will talk about the NIDDK Central Repository.
So the goals for today are to, give you a quick background of the NIDDK Central Repository so that you can have a good background of our mission, our purpose, what resources we have available, and how you can locate them, how you can access them, and more importantly, how can this be helpful to you for secondary uses. What we have is an international resource, and some of the things that Dr. Brunskill mentioned is our policy is that the repository is an international resource that supports NIDDK active and concluded studies. Those are large clinical, mostly funded through cooperative agreements. They are in the research areas that are for NIDDK's mission. And some of them are the ones that he mentioned that are funded through cooperative agreements. And most of them are large that have been from many years. And one of the things that we want to mention is that they are not only from the mission areas, but they are let me get my thoughts together. The repository program not only supports the initiatives, but also supports the requesters, right? And the repository program was established in 2003 to facilitate the sharing of the resources generated by the large consortia for secondary uses, to encourage the innovative research for investigators not involved with the originating studies and maximizing the impact to the scientific value of the resources. And although we have supported the NIDDK-funded consortia groups and the wider research community only for about 19 years, the study collections that we have in our archives have a more larger time span Some of them that have specimens and data collected over 30 years. This is an extremely valuable resource, and we will speak about their strength and applicability to you and your studies in the coming slides. By the numbers, we have serviced more than 800 investigators around the globe and fulfilled more than 3,000 requests since we began providing access 17 years ago. Our archive has more than 2,200 collections. More than 70% of those are available to the external request, representing over 150,000 unique participants, over 6 million specimens linked to well-annotated and curated clinical data. And we provide an opportunity to pool specimens across several studies to increase statistical analysis and with well-characterized and matched resources. What this does, the way that we are able to accomplish all of this, is that our program is supported by 2 separate and well-integrated contract components. That work comes together with 2 separate components that are contracted, that we provide support to program staff and external users through the life cycle of the projects. And the level of support that we provide depends really on the needs and the stage of the individual projects themselves. One of our primary goals is assisting both the submitters, meaning the consortia groups that we support, in developing materials that adhere to best practices and abides by NIH and NIDDK policies, while ensuring that we capture the intents of those projects for future research use. And we also serve as honest brokers and have dedicated staff to receive and distribute data and biospecimens, and then coordinate their operations to lead the overall operations of the team. And Mike, who's here with me, will provide additional details on how we can help you request those resources and provide additional support through the entire life of your project when you request those resources. I encourage all of you to visit our website. We include a QR code here so that you can see what resources we have available. This will take you to our main page, and Mike will speak more about what services and features, characteristics of our website, and how to submit a request as well. Mike?
Thank you, Rebecca. As Rebecca mentioned, we provide a wide range of services when you come to the NIDDK CR website. And what's critical to note here is that we assist during project planning stages before data is collected. So really, we're there throughout the entire life cycle, from data request to data curation to biospecimen request as well. Additionally, what's important to note is that the data that is being shared on the website goes through a rigorous verification and validation process. So it is, as we acquire it, it's curated, maintained, and distributed at different project stages. As I also mentioned, we do fulfill requests for internal, ancillary, and the external scientific research community, and provide support for the overall request submission process. Rebecca covered a number of the features of the data that's already stored within the NIDDK central repository. And it's also important to note that the repository makes reference to and provides direct links to other data types, as well as the reciprocal references as well. So what's nice is that you can, start out at the central repository webpage and branch out to other websites with different types of data, whether it's import, PRIDE, and we've recently linked up with Vivly, which is a generalist repository for connectivity. Next slide, please. So I won't go into any detail about the system characteristics other than to say it is a cloud-based platform. It is independent of the ERA commons for registration, authentication, and verification. As I already mentioned, that there is a verification and curation process before resources, both data and biospecimen, are released for reuse. And there is version control citable data. And we're currently in the process of implementing a digital object identifier. So that really becomes important in terms of being able to cite the data that's being used in your research. The request process has been streamlined. It's via our platform. The resources, again, are searchable through Vivly. It is electronic DAAC free review and approvals. There is automated messaging throughout and automated DocuSign features. So, this is, again, speaking to the ability to really streamline the process when you're looking to request resources, be it data or biospecimens from the website. And lastly, there is a peer review for non-renewable resources. I will say, lastly, for those of you that are familiar with NIH's Office of Data Science Strategy, the data and the platform is being built and maintained in a fair and trust compliant manner. These are characteristics that essentially make data findable, accessible, interoperable, and reusable. So there are characteristics that we and tenets that we follow, as I mentioned, in terms of storing the data and maintaining the platform. Next slide, please. So this slide, again, shows some characteristics of some of the controlled access resources available via the central repository, with several of the recently posted resources shown on the left. You can see a number of different networks and studies that are now available for release. And then you can also see, a graph on the right-hand side, which really just breaks down by observational versus clinical trial studies, and the number of data only, as well as the number of data and the associated biospecimens that are available, breaking down from 2005 to today, to 2022. Next slide, please. So I just wanted to, for the next part of this talk, I want to go through just where to locate the resources and how to access. So on our next slide, this again shows the point that both Rebecca and I have been making, that the NIDDK-CR really is at the hub of a broader data ecosystem, where we also have genomic data stored in dbGaP. We have proteomics via the PRIDE archive, and then we're showing metadata that links out to a number of different resources by which you can navigate either to or from to get at the NIDDK data and biospecimens. I will note that box on the bottom is more than just metadata. We do have clinical data that's stored in import. We've registered now with schema.org, so that if you use the Google dataset search, the NIDDK-CR will show up. So it's just, again, making the point that it has, the NIDDK-CR is a resource that has heterogeneous data types and links to a number of different repositories and search engines in order for you to navigate to the relevant data that you're looking for. Next slide, please. So the next few slides are screenshots that show you what, give you a little bit of a taste of what the R4R looks like when you navigate to the page. And first and foremost, when you come to the home screen, you can search without having to log in. So you are able to search on the metadata that describes the data sets that are stored within the repository, as well as the biospecimens. So this is the homepage that you'd land on when you go to the NIDDK-CR. Next slide, please. And if you do want to request data or biospecimens, this is where you would have to be registered and have a login. But what you'll see in the next few screenshots is really the streamlined request access review process that takes at most 2 weeks or less to return with approval. So as you can see in the next few slides, this just kind of steps you through what that process looks like. As much as possible, we try to minimize free text and employee dropdowns, and that just gives you a flavor of what that request process looks like. So with that, I will turn it back over to Rebecca. Thank you.
Well, thanks, Mike. So every request, when it's approved, let me turn on my camera back on. Every request that is approved and is authorized to be authorized for release will be or must be accompanied by a research project plan that it has a specific hypothesis or research question. It must have sufficient detail for NIDDK assigned reviewers to make a decision on the release of those resources. This will ensure that the proposed project aligns with the consent for secondary uses and that it is feasible to be completed with the resources that we have on the archive collection. The requesters will also need to submit an ethics determination for the specific project. If the review is favorable and the resources are to be released, users will also be required to sign an agreement before those resources are released. And the initial approval will be for about a period of 2 years, and it may be extended. And the repository will also make public the research project demographics that includes the investigator's name, their affiliation, the project title, along with a short paragraph that describes the project itself in layman's terms. When it comes to specimen request, we look to continuously improve access to the repository's held resources, and we have streamlined the request process to encourage utilization. In 2019, as part of this initiative, we have implemented an internal administrative review pathway for non-renewable biospecimens. Now, with that, any request that is deemed to be modest impact to the remaining collection will be going through expedited sort of is an administrative review pathway in lieu of an EXO-1 application. Those that are less abundant will go through a peer review pathway that will look at the impact to the collection. Investigators that have funds already will need to have or should submit for an EXO-1 as an alternative. Requesters interested in biospecimens should bear in mind the application cycles that I have included an example here with the earlier start dates. So in the event that the application is rerouted through the EXO-1 so that they don't miss the cycle timelines. For example, if you were interested in submission or to start your project in April 2023, you just missed the timeline. So if you submit right now and your application was deemed to be eligible for an administrative path, but was rerouted to a peer review, you just missed the cycle and it will not be reviewed by a peer review until June, and you will not be able to start your fulfillment process until July. So you have to bear those things in mind. We include here a QR code to the PAR for the EXO-1 so that you can get more familiar, more information about the application process. So how can these resources be helpful for you? And the best way to tell that is with some success stories. What some success stories look like, and the best, sometimes all we have is publication rates. As we mentioned in these introduction slides, we have granted access to more than 800 investigators over the years and fulfilled more than 3,000 requests. Just in the last 5 years, we have fulfilled more than 530 requests for access and received 91 pre-publication clearances. These are public releases. The number of public release or publications is likely underestimated, and this only accounts for those that submit pre-publication. We don't know exactly how many are those that forget to submit their manuscripts to us. So this swim plot shows the top requested studies and against those that submitted the publication clearance in the 5 years. And you can see that we do get quite a bit. One of the examples that we have on the top stories is Dr. Terera, for example, is an early stage investigator such as yourselves who requested data from the Look Ahead study. This study examined the lifestyle intervention and the prevention of cardiovascular disease in individuals with type 2 diabetes. And Dr. Terera performed a secondary analysis on the Look Ahead, looking at cardiovascular risk factors with sex-specific differences between fat mass and muscle mass. Terera findings were that A1C may predict future A1C cardiovascular disease and suggest that maybe the strategies to lower A1C may be of importance, especially for females with low fat mass and low muscle mass who are living with type 2 diabetes. For sake of timing, I'm not going to go through all the examples that we have, and we can share this with you, but one important success story is Dr. Teresa Chen, who is one of the few EXO-1s that progressed to a K award, which is a very good success story. Dr. Chen evaluated is an early stage investigator who used the African American study of kidney disease and hypertension. She evaluated examined immune biomarkers and the developmental progression of chronic and end kidney disease, more specifically the APOL1 risk variants, which is a genetic risk factor for kidney disease among persons of African ancestry. One of the things that she found is that there's no association between the interferon gamma and APOL1 high-risk status, once thought to be related, and proposes that this baseline biomarker may not be as biologically relevant in APOL1 mediated kidney disease among the African ancestry persons. So I encourage you to look at the resources that we have so that this may help you in your secondary study analysis. Besides the obvious, of course, secondary data analysis, save time and money and are good. But what can these help you with is, the use of secondary data analysis is that saves you time and money. But these resources that we have within the NIDDK archive have been expertly curated. What Dr. Brunskill mentioned earlier is that they expand large years, and it's unlikely that you will be able to duplicate this in any time soon. These have been backed by NIDDK. They have been supported with funds, and programmatically they have been supported by NIDDK. We have rich study data, study metadata, and we have good documentation. So it is unlikely that you could find this elsewhere. So I encourage you to think about what you could do with this secondary analysis.
I would like to quickly introduce Dr. Martone. She has a bachelor's from Wellesley College in biological psychology and a PhD in neuroscience from the University of California. She's currently a professor in the Department of Neuroscience with a background in neuroanatomy. She will be talking to us about the dkNET. I did want to mention that people from the dkNET will be present at the CAIRIBU annual meeting where they can discuss in further detail the resources that are available there. So, Dr. Martone.
Thank you very much for the opportunity to talk about the NIDDK Information Network, or dkNET. I do have a disclosure that I'm a founder and board member of SciCrunch Inc. You'll see the word SciCrunch throughout dkNET, that builds on services around research resource identifiers, or RIDs, which I will be talking about today. So I wanted to make sure that I was transparent. So first of all, what is dkNET? Well, you've already seen that there is a tremendous wealth of resources that are available to you as young researchers that are both funded by NIDDK. There are many more that are available that are not funded by NIDDK. And I think, as also was alluded to in the introduction, it is not always easy to be aware of the breadth of these resources. So at its heart, dkNET is a search portal that provides a single point of access to information about these diverse research resources, which includes data, information, materials, tools, funding opportunities, et cetera. But as you'll see, we also have many other products that are and services that are really relevant to researchers starting out. And that includes support for some of the NIH policies on rigor and reproducibility. And you've already heard the FAIR data principles mentioned. If you're starting out your career and you haven't heard of it yet, you will hear of it a lot, because the NIDDK, the NIH data sharing policy, of course, is requiring that all data be fair. And I was actually one of the authors on the FAIR paper, and dkNET is providing a lot of educational materials, training opportunities to understand what FAIR is and how it is going to impact your career in the future. So we have a lot of information. We are an information network. Organizing that is always hard, and we're constantly trying to improve the way that our portal works. But I thought it would be interesting just to break down, as I just mentioned very quickly, the types of things that dkNET is supporting. So the first is findability, rigor, and reproducibility of research resources, antibodies, data, tools, all of those things. We have several sections of our portal that deal with that, and I'll go over something called resource reports in just a bit. We also, though, have a program that is designed to increase the computational skills amongst the NIDDK workforce. So we have introductory materials on how to use a lot of these bioinformatics tools if you are not a bioinformatician, called the Hypothesis Center. We actually support a new investigator pilot program in bioinformatics. We've had multiple years of young investigators receiving pilot awards. We don't have the information about next year's program yet, but that will be up on the website as soon as we know. And we also host a webinar series that feature a lot of the types of tools that you've heard about already. And you will also see, as you go through dkNET, these webinars are posted on our website so they become a resource for how you can actually learn to use a lot of these tools. And as I mentioned, we're trying to bring fair and open data practices to DK researchers. We hold webinar series. We have a listing of repositories for data sharing. We provide help on data management and sharing plans. And I'll mention a little bit about our Summer of Data student program, which I know PIs at the CAIRIBU program have sent us students in the past that really help train the next generation workforce on these important topics. So the overall portal is organized into 5 main areas: resource reports, authentication reports, which kind of go together, FAIR data services, the discovery portal, and the Hypothesis Center. If you think about and start to use all of the tools that we have, you realize that we cover the entire life cycle of being able to do background research, to formulate a hypothesis, to constructing it, to planning experiments, getting reagents you need, collecting and analyzing data, and finally publishing results. So of course, this is very ambitious, and how we organize this is always a challenge for us, as I mentioned. But if you go to the dkNET homepage, we've tried to organize the entry into these tools into 4 main areas: resource reports, authentication and fair data, the discovery portal, which searches across biomedical databases inside of these databases. So the NIDDK Central Repository, for example, is there, and the Hypothesis Center, which I mentioned already. However, if you go up to the About menu, you will see some of the other services that we have that I just mentioned. So for example, here, this is where you would find our bioinformatics program that I mentioned, also our summer program you can access from there. And we will be announcing the 2023 program soon. This is a program to send students to learn training, not in necessarily data science, but all the precursors to data science, which is how do you manage your data? How do you share it? How do you make it fair? What is fair? So it's been a very, very successful program. And we hope that we will continue to see members of your community take advantage of it. One other interesting resource that we have is a lot of the centers that NIDDK funds actually have little pilot programs that are available to you. And we didn't see any database that covered these types of pilot opportunities. So we routinely work with the centers, go around and look at the web and try to pull these together into a searchable database so that if one of these may meet your needs, you can go there, look at it, and hopefully apply for these. Many of these pilot programs give you small amounts of seed funding that then can be parlayed into larger grants. And one of the things we've introduced recently, and in fact, the urology community is our first example, is to try to make it a little easier for people who are just starting out to understand what it is that dkNET has available. We're creating these special collections around topic areas. So if you go up to the About menu again, you'll see that, or look at our homepage banner, there is now a urology collection where we've gone through and we give you tutorials, we give you specific examples, and we do sort of pre-filtered queries for you so that you can kind of understand what it is that dkNET does. And of course, we are always happy to answer questions. If you can't find anything, please contact us, because we can often pull together the resources that you need. So I didn't want to go into all of the things that dkNET does, because there's a lot of them and we don't really have time. But I did want to focus a little bit about resource reports, because this is really where dkNET has assembled a very, very unique and I think useful data set that will allow you to understand both the breadth of resources that are available, but also give you some tools for perhaps evaluating them and how good they might be for your research. So resource reports, I already mentioned, is available through the homepage. If you click on it, you'll see that there are different types of resources that we provide. Tools, which are really digital tools that you find online, software tools, databases, data sets, services, training programs. It's a very large catalog. You can see over here, there's over 20,000 of these resources that we have. And if there's anything that we don't have, you can add it. So if you are producing tools yourself and you don't see them in our catalog, we make it easy for you to go ahead and add your resource so that it becomes available. Antibodies, we maintain the antibody registry, which has over 2 million antibodies constantly being added to all the time. Plasmids via AdGene, and also the Drosophila community. Protocols via protocols.io. Cell lines come via Cellosaurus. Organism actually indexes over 25, I think 30 different sources that provide access to model organisms and other types of organisms funded by the NIH Stock Center. So it's a very, very rich data set. You see it's got over 600,000 resources, and also biosamples. So we work with several programs funded by NIDDK, including the islet cell program, the IIDP, and you can both search these biosamples. And as I'll show you in a minute, these resources that you have here, this catalog, is really the source for these research resource identifiers that I mentioned. But first, I want to go into a little bit of anatomy of a resource report. So we provide basic metadata information. So of course, the idea is to put you in touch with these resources so that you can obtain them for your laboratory. Some of them may require costs. Most of these are free beyond shipping and other sort of minimal costs. But we don't just provide that metadata. We also, through these RRIDs that I'll mention in just a moment, and other tools that we deploy, we search for the usage of these tools across the biomedical literature, most specifically the open access biomedical literature, but millions and millions of articles, to see who else is using these. If you click on our collaborator network and you put in your location, you can find authors on these papers who might be near you, or at your own institution, that have experience working with these resources. It's very useful. And we're also aggregating information about validations or performance issues that have been reported by the centers that often have panels of antibodies that they recommend, for example. So we aggregate this information and we make that available to you, and other independent entities who are working to validate or uncover issues with these. So if any of these happened, we happen to find these, we distribute these through the resource report, so you have some understanding of how it is that it performs. If you click on our mentions, it takes you to this page. It'll give you a listing of all of the papers that we found that mention that particular resource, and also if other resources have been used in conjunction with these that we recognize, we'll pull them out and we'll let you know. So oftentimes, for example, if there's a particular data set, you'll find a list of software tools that are often used with that data set. So it's a very, I think, rich and useful set of information that I don't think is replicated anywhere else. And as I mentioned, the reason why we were able to assemble this really unique data set is due to something called the Research Resource Identifier, or RRID. If you publish in endocrine society journals, you will be asked to supply these research resource identifiers. These are these unique identifiers, just like the DOI that you heard mentioned before, only these are applied to all of these different types of research resources that we talk about: antibodies, plasmids, digital tools, etc. And because we have these unique numbers that are being used in the literature by authors, this is supported by over 1,700 different journals. All of the major journals now require RRIDs. So using our literature mining tools and other resources that we have, we're able to pull these mentions out. Antibody companies and centers, NIDDK-funded centers, use that RRID. So that makes it easy for us to pull this information together in something that we call the Resource Identification Network, or RIN. That's really just the data set that underlies dkNET's resource reports. So as we mentioned, it's a persistent identifier for research resources. dkNET was instrumental in the development and the adoption of RRIDs. And the reason that we developed the entire program actually was because when researchers were publishing their papers, they were not putting enough identifiable information into their papers for you to know exactly what resource they used. So for example, over 50% of the antibodies in published papers before the introduction of RRIDs didn't tell you the catalog number, didn't tell you the vendor, so you had no idea what antibody was used. But also, as you saw already, because we have this unique identifier that really simplifies and makes search more accurate, we can also ask that important question of who else uses these research resources. So it becomes a tracking mechanism for research resources in the scientific literature, which is not anything that we had before, but is so important. Why is that important? Because we know that there are problems with these resources. We know there are specificity problems with antibodies. We know that cell lines are contaminated. And because this information was scattered across hundreds and thousands of information sources, nobody knew about them. But now, thanks to really dkNET, we aggregate all of these issues. When authors go to put these resources in their papers, they are confronted with the fact that the cell line that they used may be problematic. And so we're really encouraging people to come to dkNET before they do their research and before they plan their research, and to sign up for our alerting services, because this way you know before you go to publish the paper and not afterwards. So I wanted to just spend a few minutes on how you actually search the catalog, because as we mentioned, all of these resources, they take a little bit of time to learn. Ko-Wei Lin is our project manager, and she's on this call, and she is a whiz at this, and she's also always happy to help you if you run into any troubles. But basically, we allow you to search through free text. And if you type in something in free text, we have a huge database of terminology behind that. So we'll automatically search for synonyms for you. That often helps uncover things that you wouldn't get otherwise. Every time you do a search, you get a set of facets that are specific for that type of resource. So this is an antibody, so you have relevant metadata here that lets you further refine your search. But we also always provide this mentions facet here. So this is access to that literature mentions that we talked about, and also issues. So if there's been any issues that have been reported, this is what lets you actually find that out. You can download results once you find them. You can, as I mentioned, create collections. You can follow these research resources so that if any new information comes up, you are alerted. So it's a very nice system. You can either look at things as a snippet, or there's also a table view, which shows you the whole thing in tabular form. And I just wanted to show you how you can use some of these filters to answer a question, for example, like find the most popular validated antibodies against phospho-ATK, AKT, sorry, I'm having trouble reading this, it's a little small, probably small for you too, that are in use in the literature. So all you do is you do your search for your AKT, you then come down and say, I'd like to only see antibodies that have literature mentions, and I'd only like to see antibodies that have validation information associated with them. That brings up the list of antibodies that satisfy that criteria. You can see that this phospho-AKT has over 100 mentions in the literature, so it's very, very popular. It also has rating and validation information available from the intestinal stem cell consortium, and if you go there, you can find that, and then you get this list of papers that have reported the RRID for this, so that you can be sure that this information is accurate. So I think you can see that by learning how to use dkNET, you can actually really delve into a very rich and unique source of information to help you plan your experiments. I mentioned the authentication reports a bit. NIH actually requires you to submit a report on how you're going to validate your antibodies. Getting RRIDs and looking at information on how these resources are supposed to be used and any issues associated with them can go into that report. And we have a little tool called the authentication report that provides various guidelines about this, lets you put in your reagents and create a nice snapshot report of anything that we know about them, recommended uses, those sorts of things. So I encourage you to explore that. Okay. So I'm going to just wrap up now. I know we're a little bit late, but we have several means of communications that you can use to keep up to date about what's going on in dkNET. We mentioned our webinar series. We have a newsletter that comes out. You can sign up for our mailing list. And that's a very good way to get a digest every month of new resources that come out, new opportunities, new funding opportunities. So we encourage you to do that. Also our webinar series and any other tools and things that we think are useful to you. And so I hope that if you have any questions as you start your careers and you progress about research resources, anything in this whole landscape, that you ask dkNET. Thank you.